I'm Professor Joshua Sharp of the University of Mississippi, and today I'm going to tell you about work that we've been doing in collaboration with Professor Lisa Jones at University of California, San Diego, developing new technology to bring radical protein footprinting to mammalian whole blood samples. These include preclinical and hopefully eventually clinical samples. To give you a little bit of background on structural proteomics by fast photochemical oxidation of proteins, or FPOP, hydroxyl radical protein footprinting has a long history of being used in vitro, spanning a couple decades now. And it's posted a lot of successes in terms of looking at the in vitro structure of proteins, their conformational changes, the complexes that they form. And in 2015, Lisa Jones and collaborators published the first example of actually applying FPOP to look at protein conformation inside of live cells. And since 2015, her group has expanded this technology to look at protein structure in live nematodes, and now most recently in 3D cell cultures, such as solid tumor spheroids. All trying to move the technology into more and more biologically and clinically relevant conditions. However, kind of the ultimate goal of looking at protein structure inside of living mammalian tissues is still out of reach. And the major reason that it's out of reach is because of the strong tissue absorbance in the UV. Since FPOP and related measures of hydroxyl radical protein footprinting require UV light to activate the precursor chemical to form the radicals needed to do the labeling, the inability of UV to penetrate mammalian tissues to any appreciable depth has prevented its use inside of mammalian tissues or living mammalian organisms. Just to give a brief update, if this is your first experience with hydroxyl radical protein footprinting, briefly, the way that it works is you have a protein, and the most common way people use it is they look at a protein in different conformations. In this case, we've got a ligand-bound version, and we've got a ligand-free version, and you expose that protein to some form of radical that you generate in situ that can diffuse through solution to the surface of your protein. And this radical will react with the amino acid side chains in your protein to generate these sites of modification. And the sites of modification and the extent of modification at each site will be proportional to the exposure of that site to the radical and therefore to the solvent itself. Once you've made these modifications are stable. And so you can then denature your protein, digest it with your favorite protease, and come up with a collection of modified peptides in which the pattern of modification that originally existed in the protein is still captured in the pattern of modifications of the peptides. You can then measure the extent of these modifications using LC-MS and LC-MS-MS to measure the amount of modification for each peptide in each of the different conditions that you're measuring, and measure which peptides, for example, show changes in oxidation from one condition to another that therefore reflect changes in protein solvent accessible surface area. And then from these data, you can go on and start to look at things like find ligand binding sites, find conformational changes, things of that nature. So it's a very powerful way to look at solution phase topography, surfaces of proteins, and how they change under different conditions. Our goal in this collaborative work is to generate the first method for looking at protein structure inside of a mammalian tissue, and the tissue that we are targeting is mammalian whole blood. In blood, FPOP would have major implications in terms of structural pharmacology, being able to look at protein structure and protein interactions in the blood or potentially within other human fluids. Things like anti-drug antibody interactions to identify anti-drug antibody epitopes inside on biologics, drug target interactions, drug off-target interactions, and even things like post-administration aggregation pathways to help for the discovery and development of better biologics. In addition, there are more general applications for structural proteomics in human fluids, things like liquid tumors, things like biology and diseases of blood, as well as of other potentially biological fluids in the future, such as lymph, cerebrospinal fluid, etc. And these are not minor problems. So the problem that my lab is really interested in is looking at anti-drug antibodies. And anti-drug antibodies for some particular therapeutic modalities is a major issue. So for example, in this review, they looked at oncology checkpoint inhibitor therapies, and they found that in the patients that they looked at, the percentage of patients that developed anti-drug antibodies for some treatments was as high as 50%. And so that's a major issue in terms of patient safety and treatment efficacy. Understanding which parts of the drug these anti-drug antibodies are targeting could really help to in the design and development of better drugs that show a little bit more patient immunotolerance. So what's the big challenge for developing FPOP in blood? And the number one challenge for developing FPOP in blood is not the UV absorbance issue, because since it's a liquid, we can actually look at the fluid in relatively narrow path lengths to where we can get enough absorbance of the UV. The major issue for doing FPOP in blood is catalase. Catalase is a very, very robust and highly efficient enzyme that catalyzes the degradation of hydrogen peroxide into water and oxygen. And for traditional FPOP, hydrogen peroxide is the radical precursor that's photoactivated to generate the hydroxyl radicals. So when you try to do FPOP in blood, you add the hydrogen peroxide to the blood, and the blood immediately, the catalase in the blood immediately degrades the hydrogen peroxide to where you have no radical precursor left. There are other issues, but these issues are issues that exist in other kinds of in-cell FPOP, things like proteomic complexity. Blood does have a very, very exaggerated dynamic range issue where the most abundant proteins have a much, much higher abundance than the least abundant proteins. So these are issues that will pose problems in FPOP. But the number one issue we have to deal with is the catalase. How do we do FPOP in the presence of all of this catalase activity? Our first efforts we were trying to find an inhibitor for catalase that would allow us to inhibit the enzyme, but still maintain the gross properties of the blood so that we could be reasonably sure that we're looking at native protein structures and complexes. We tried 4 different inhibitors, hydroxylamine, aminoguanidine, TSC, and 3-AT. And we found that for none of the inhibitors at any of the concentrations that we tested were we able to completely inhibit the catalase activity without grossly altering the properties of the blood. So I'm showing here the results from the hydroxylamine. And you can see as we increase the hydroxylamine concentration, we're measuring catalase activity by a very simple assay known as a foam assay. In the foam assay, what you do is you take the blood, you add a little bit of very, very mild detergent, and then you add in your hydrogen peroxide. And if the catalase is active, the hydrogen peroxide will be degraded, it will form bubbles, and the bubbles will be captured in the detergent and form a layer of foam on top of your blood. You measure the height of the foam layer, and that gives you a relative measurement of the catalase activity in your blood. And as you can see, when we add in hydroxylamine, we don't see an elimination of this foam layer, even at very high concentrations that are high enough to change the color of the blood in a very noticeable fashion. It turns this dark, dark kind of purple-black. And you end up with a blood sample that is nowhere near to biologically relevant. And this was true for all of the different catalase inhibitors that we tried. We couldn't find any inhibitor that would actually inhibit catalase activity without completely altering the properties of the blood. So peroxide was not going to work. Luckily, peroxide is not the only precursor that can be activated to generate hydroxyl radicals. So we decided to try a separate precursor, and this precursor was sodium persulfate. So fortunately, when we tried sodium persulfate, we add it to blood with no inhibitor. In the foam assay, we see absolutely no foam generated. The catalase or any of the other peroxidases or other enzymes in the blood do not break down the sodium persulfate, do not form the bubbles. And so we are going to be focusing on using sodium persulfate as our radical precursor for FPOP. So then the next question is, how do we activate our sodium persulfate? All of the previous work has been done with lasers. And while lasers are very, very useful and have a long history in hydroxyl radical protein footprinting, they're not particularly user-friendly. They're very difficult to approach, and they're not particularly convenient to work with. So we wanted to try and see if we could get persulfate oxidation to work using the phosphotolysis system, which is manufactured and sold by GenX Technologies. The phosphotolysis system is able to perform FPOP, but it uses a flash lamp, an ultra-fast flash lamp system. And so it's much safer than lasers. It's a lot more convenient than lasers. And so the first thing we need to do is see, can we actually get persulfate-based oxidation using a FOX system? And so we started just doing standard adenine dosimetry, looking at, can we actually see a difference in adenine absorbance using persulfate instead of hydrogen peroxide? And what we can see is that, yes, we are actually able, using persulfate and the FOX system, we are able to oxidize our adenine dosimeter very efficiently. And this oxidation is inhibited by our standard hydroxyl radical scavengers like sucrose. So as we increase our lamp voltage with persulfate, the absorbance of our dosimeter decreases, as you would expect. As it becomes oxidized, the absorbance should decrease. And then at a fixed concentration of persulfate and a fixed lamp voltage, as we increase our concentration of radical scavenger, our absorbance goes back up, because the adenine dosimeter is seeing less radical. It's being scavenged by the sucrose. This is exactly what we would expect. And so not only does persulfate oxidation work, our standard dosimeters work in vitro. So you can do persulfate-based oxidation using the FOX system. However, this dosimetry method does not work in blood, and that's because the background absorbance at 265 nanometers is far, far too high. So that's another issue we're going to have to deal with at a later date, is how do we do real-time dosimetry for our in-blood oxidation. But for now, we know persulfate oxidation does work with the FOX system. So we have our precursor, we have our light source. The next thing we want to look at is the quench system. So after you generate your radicals, the radicals themselves self-quench. However, they also generate secondary oxidants. And these secondary oxidants, along with leftover radical precursor, can oxidize your proteins, albeit much more slowly than the radical-based oxidation. And this background oxidation can actually interfere with the results that you would get from your FPOP experiment. So it's very important that you have a very efficient quenching method in order to eliminate that background oxidation. Quenching for peroxide-based FPOP has been very well explored. There's a very robust method for quenching that uses dimethylthiourea and methionine. However, when we move to persulfate instead of peroxide, and we try this same DMTU methionine quench, you have some peptides shown here in gray that still show an elevated level of oxidation compared to the no persulfate control. So this is 2 through 17 peptide and this 120 to 134 peptide show elevated levels of oxidation. And so this is consistent with what had previously been reported from persulfate-based modification from the Jones lab, in that they did also notice an elevated background in persulfate compared to hydrogen peroxide when using the standard FPOP quench. So we tested a few additives to the quench to see if we could eliminate this elevated background for these peptides. And one that we found that worked well was adding 200 millimolar imidazole to the DMTU methionine quench. When we did that, this background oxidation decreased to 0. So the amount of oxidation we see with the persulfate equals the amount of oxidation we see without persulfate for every peptide. Hydroquinone and reduced glutathione also work, but they have other issues, so we are going to stick with imidazole here. So we have our radical precursor, we have our light source, we have our quench. Now we need to know, does our method actually kill all of our red blood cells before we can actually probe protein structure? Because again, we're trying to look at protein structure in a biologically relevant or clinically relevant context. And so what we did is we took a look by microscopy at blood cells by themselves, and then what happens when we mix the blood cells with sodium persulfate. So this is just standard bright field microscopy. No dyes were used. Any difference you see in color is just a difference in contrast. And what we see when we look, normally you see, you get a lot of cells that look, spherical. You see some of these cells that have these spikes. This is evidence of hypertonicity, hypertonic conditions. But you don't see a whole lot of ghost cells. You don't see a whole lot of cell debris. This is what you would expect from a blood sample that has been stored relatively well. When we take this blood sample and we add in 200 millimolar sodium persulfate, which is the concentration of persulfate we're going to be using, and we look, you see there's not a whole lot of changes in the gross morphology of these blood cells. You do see a modest, small increase in the percentage of cells that show symptoms of modest hypertonicity. So the elevated salt concentration is causing some hypertonicity in these cells. But you don't see a lot of ghost cells. You don't see a lot of cellular debris. The cells look relatively healthy. And just to verify that this hypertonicity is just due to changes in the salt concentration, we replaced the sodium persulfate with sodium chloride with the same concentration of sodium chloride and looked, and the hypertonicity is even more acute here with the sodium chloride, as one would expect. But we don't see any cell morphology here in sodium persulfate that we don't see in sodium chloride. So this does appear to be just a hypertonicity issue, and not an issue of anything else that's going on in terms of the sodium persulfate. So the gross morphology of the cells suggests that we are looking at biologically relevant sample here, and so we should be ready to go. So here is the in-blood FPOP system that you can see hooked up to a GenX phosphotolysis system here. Briefly, what we have two syringes. In one syringe is connected via a 3-port manual switching valve to a reservoir that contains your blood. The other syringe is connected via a 3-port manual valve to a reservoir that contains your sodium persulfate. This is a 500 microliter syringe, is a 50 microliter syringe. And so there is a 10-to-1 volumetric difference between these syringes as they are driven by the pump. And so you start with your withdrawal step where you pull both the blood and the persulfate from the reservoir into the syringe. You then switch these switching valves and then push the sample out, which flows through these fused silica capillaries to a mixing tea. The samples are mixed 10-to-1 in the mixing tea and then pushed up to where they go into the photolysis pathway of the phosphotolysis system, where the persulfate is photolysed to generate your radicals. So that's the fluidic system. And so the first question we had was the mixing in the mixing tea, right? So we've got a 10-to-1 volumetric difference that we're trying to mix in the mixing tea. And the question we had was, how long does it take for these volumes to evenly mix across the capillary? Because we want an even mixture of the persulfate and into the blood when we actually photolyze it. So what we did is we took two dyes. We took a blue dye that we put into the blood syringe and a red dye that we put into the persulfate syringe. And we ran it through the mixing system, and we took bright field images of the capillaries at different distances from the tea. Okay, so 2 centimeters after mixing, 3 centimeters after mixing, 4 centimeters after mixing, et cetera, et cetera. And we looked at the distribution of color across the capillary. So you can see here, if you look very closely, this is just looking at the red dye channel from the bright field image. You can see you've got this dark band across the top of the capillary, which is the part of the capillary closest to where the persulfate would come in. And then at the bottom of the capillary, you see much, much less of the red color. As you get 3 centimeters from the T, you see this dark band becomes more distributed across the whole capillary. At 4 centimeters, it's even more distributed, and we can actually measure this across a cross section of the capillary. So at 2 centimeters, you see there is a broad distribution between lightly colored and darkly colored regions in the capillary. As we get 3 centimeters away, this distribution contracts as these two start to mix more. And by the time you get 4 centimeters away, you have a nice unimodal distribution that shows even mixing across the entire capillary. So once we get 4 centimeters from the T, we are evenly mixed. So for all of the samples that I'm going to show you today, flash was done about 5 to 6 centimeters from the T. So well beyond the even mixing range. And so we took our blood, we mixed it with a persulfate using the fluidic system. We flashed it at 900 volts. And then we did proteomics, we did LCM, SMS, after tryptic digestion to measure the oxidation of different proteins. And so the first protein I'm going to show you is hemoglobin. So this is hemoglobin, as you can imagine. When you look at whole red blood, this is the #1 protein that you see, is hemoglobin. Shown in the different colors here, the different shades of red is different peptides that we observe as oxidized, and with the shade of red correlating with different levels of oxidation. And you can see, we see the vast majority of this protein in some form of oxidation. So our oxidation coverage is actually very, very high. The only peptides we don't see oxidized are colored here in green for alpha globin, or light orange there for beta globin. But the vast majority of the protein is well oxidized. So that's really excellent oxidation coverage. So we're seeing really good oxidation, and we're able to detect different levels of oxidation for different peptides. But how do we know that this is actually reflective of the folded structure of hemoglobin and not just random? So to do that, what we did is we took isolated hemoglobin and we ran it in vitro, so just in test tubes, and looked at the amount of oxidation that we got for all of the different peptides there. And this has been well established in vitro that the amount of oxidation is proportional to the solvent accessibility of that peptide, with some other caveats, but that's the vast majority. And what we want to do is see how well does the amount of oxidation we see in whole blood correlate with the amount of oxidation we'd see of the same peptide in vitro isolated. So we did that experiment. We had to do, so the blood that I'm showing you is mouse blood, whereas the hemoglobin, the isolated hemoglobin we have is bovine, because it was very difficult for us to source isolated mouse hemoglobin, but the two are 74% identical, 86% similar. So we're able to look at peptides that are either identical or very, very highly homologous. And what we did was we took every peptide that was oxidized that had a homolog in the other version of hemoglobin. So every peptide in mouse that was oxidized that had a homolog in bovine hemoglobin, and we plotted the amount of oxidation. The amount of oxidation in vitro is on the x-axis, the amount of oxidation ex vivo in the whole blood is on the y-axis, and what you can see is we get a beautiful correlation. Peptides that are highly oxidized in vitro are also highly oxidized in blood. Poorly oxidized in vitro or poorly oxidized in blood, it goes across the entire range. Our R-square value is very, very nice. So we are definitely oxidizing blood proteins in the blood, and this oxidation is reflective of the solvent accessibility of that protein. So we are getting biologically relevant data out of this. Of course, hemoglobin is not the only protein that we saw, so I'll just show you a few more. Here's albumin. Our sequence coverage of albumin is not great, but for the parts of albumin that we do see, we actually get very nice oxidative coverage. We're looking to see why we're missing parts of albumin. We suspect it might be because of all of the post-translational modifications you can get on albumin that we're not accounting for. But we are seeing that when we see the peptide, there's a very good likelihood that we have oxidized it. So our oxidation coverage still looks pretty good. This is serine protease inhibitor. Very good sequence coverage. Oxidation coverage is a little bit lower, but we still have many, many oxidized peptides in this protein. Here's superoxide dismutase and apolipoprotein 4, two more proteins that we were able to identify multiple oxidized peptides from these proteins. So what we're working on now, we're working on moving from manual fluid handling to an automated fluidics handling, so that instead of the manual valves that we have to turn and the syringe pump we have to operate, we've written custom software fluidics control. We've replaced the manual valves to where all of the fluidics handling is now handled through an automatic low volume switching valve, and we've mounted it onto the Fox system now. You can see our prototype in operation right here. And so that's kind of our next step, is to move this to a more automated technique. We're working on new dosimetry methods to try to do a better job of being able to measure the amount of radical we're delivering to our protein. We need new informatics and sample handling methods to deal with the proteomic complexity and dynamic range. And we also, for my particular interest, we need workflows to do targeted analysis so we can focus on specific proteins from whole blood to answer some of the pharmacology questions that my group has. With that, I want to thank the people that helped with this work. The funding was through the NIGMS. Want to thank Glycor, the imaging core at Glycor, along with Professor Greg Roman, for helping with the imaging of the red blood cells. GenX Technologies, which graciously loaned us a Fox photolysis system and even let me drill holes in it. So I appreciate them for that. The people that did the work, Lyle Tobin, very talented undergraduate working in my lab, who did a lot of the in vitro work and the catalase inhibition work. Dr. Sandeep Misra, who did a lot of the data analysis and some of the in vitro work. Aaron Sharp, undergraduate computer sciences major, who wrote our custom control software for our automated fluidics handling system, as well as Darian Martin and Jonathan McCaskill, who have done some work on portions of this project that I did not talk about in this particular talk today. From the Jones Research Lab, our collaborators, Professor Lisa Jones, who graciously hosted me in her lab for 6 months on my sabbatical to work on this project, as well as Howlin Liu, who helped with doing some of the proteomics out of whole blood. So thanks to them both, and thank you all for your attention.